Good evening.
Good evening, ma'am.
Evening. So let's just start. It's time. For the last class, Shamika, if you remember, we have been discussing about the gene rearrangement that takes place, and we discussed about the enzymes involved. We discussed about two specific enzymes that are only present in the lymphocytes. So these can also be called as the markers for lymphocytes. And we briefly saw something, we saw it for light chain. We discussed that it's always the kappa chain that rearranges. And if the rearrangement rendered is non-productive, the lambda chain is given a chance to rearrange. First, it's the heavy chain that undergoes rearrangement. It's in the same mechanism, only difference being that there is also a D region between the V and J region. And once the heavy chain rearrangement occurs, it's the light chain that gets a chance to rearrange first kappa, then lambda. And we also saw that there is something called, we briefly discussed very briefly that there's something called as class switching, wherein the variable region remains constant, and it is only the constant region of the heavy chain that is further subjected to some sort of rearrangement, and we have the phenomena of class switching happening. We then started with the discussion around the mechanism of all these rearrangements that we have been discussing. And today we take in detail about the mechanism, everything that is involved at the backstage, right? Okay. So we discussed that alongside the V, D, and J exons, we have certain sequences that flank these regions on the either sides, called as the recombination signal sequences, right? So these recombination signal sequences, they consist of nonamer and heptamer sequences, which are very conserved. So throughout different individual, you will find that the sequence of these nonamer and heptamer region, they do not really vary a lot, right? These two sequences can be separated by what we call as the one turn or two turn recognition recombination signal sequences, right? So one turn is corresponding to 12 base pairs of nucleotides, because the bDNA that we all have, the spacing and all is such that when there is 12 nucleotide gap, it completes one helical round. And if there are 23 base pairs in between the nonamer and heptamer sequences, the DNA actually gets a chance to complete two rounds, two turns. So that's why they are called as two turn or one turn recombination signal sequences. And we also saw that for lambda chain, we have so if you look here for lambda chain, we have two turn RSS sequence that is downstream of the variable region of lambda chain, and we have one turn sequence upstream of the J region of lambda chain. So there's a pattern to it that whichever two regions have to join together, one of them would either be having a one-turn sequence, and the other one, the next of it, which has to come and join to this previous segment, will be having a two-turn sequence. So it's always going to be opposite, right? For heavy chain, so it's not necessary that with the V region we will have a one turn or a two turn sequence, because if you see here for kappa and lambda chain, the V region downstream of it is a one turn sequence for kappa chain and two turn sequence for your lambda chain. But with respect to that, your of the kappa chain is also having a two turn sequence. So all that sort of arrangement is there to ensure that we always have the correct gene segments coming and joining to each other. This is ensured by the phenomena, by the underlying principle that it is always going to be this one turn sequence joining to a two turn sequence. You will not have a one turn sequence that can join a one turn sequence. It's always going to be opposite to each other. In the heavy chain arrangement, this thing is a little more clear to understand. The significance of it becomes more easier for us to understand because now there is a D segment in between, and on both the sides it's flanked by a one turn sequence. And here you have a two turn sequence with your variable region and a with the V region and a two-turn sequence upstream of the J region. Had it not been the case, you could have this, say, the V region coming and joining here as well as here. But this thing is prevented because of this principle that a two-turn sequence can only join to a one-turn sequence. Okay, so this much we discussed in our until our last class, and for today's class, we will look into the mechanism of it. So it is a little complex looking diagram, but we'll try to simplify and break it. If there's anything that comes, you crosses your mind until the time you are the only one in the class, we can stake the doubts simultaneously, right? You can just stop me wherever you feel you want to ask something. Okay. So before we look into it, let's just first try and understand that what happens in the overall process. So we have a RSS signal which can be recognized by the recombinase, which is the enzyme that is actually mediating this entire process. Recombinase, which is your RAG1 and RAG2, right? So what does it do? This RAG1 and RAG2, it recognizes all this RSS sequences here. We have two players in this process. So say one of it is going to recognize it. Once your RAG1 recognizes this RSS signal, it facilitates the further binding of RAG2. This RAG2 before it comes in association with RAG1, it has also recognized the RSS sequence of the other gene segment. So say that one RSS segment of the V region is recognized by RAG1. The other RSS sequence of your J region is recognized by RAG2. And ultimately, RAG1 and RAG2 needs to come together in a synapse sort of situation. The synapse is bringing of these two things together into some sort of communication, right? So once on this entire I would have. So let's just imagine that this is, so since say this is your linear DNA, the entire linear DNA, and maybe these two overhanging regions is your V region, and this might be your J region, right? This thick portion here, say this is the RSS sequence and of your V region, and this thick portion here is your RSS sequence of your J region. Now we have two enzymes that have recognized these RSS regions, and they are now going to ultimately bind to each other. So in this process of these two coming and binding to each other, what is it doing? It is bringing your V and D and the J regions together if we are talking in context of light chain. If this is in the context of heavy chain, we have the D and J joining first, and then this V segment joins the D and J segment, right? So the overall process is that these two things have now come together. We have, if you just have a look like this, so it will be one single strand of this green stretch of DNA, which becomes my coding sequence. And whatever is there in between, this thing has to be spliced off, cut off, right? So this is exactly for you to visualize, this is exactly what is happening in this entire process. We have RAG1, RAG2, which is going to bring these two segments, RSS segments in a synapse in a close proximity, and eventually one strand, firstly, there's just going to be one strand which is cleaved by the RAG1 and RAG2 at the juncture of your signal and the coding sequence. So just as we saw that at these two junctures, so this is your signal sequence, so this is the juncture of your coding and signal sequence. At this point, only at first, only one of the strand is cleaved. So once this is cleaved, this is going to say create a free end. So we will have, say, I cannot break this now, but we will have a 3' OH for one, and another way we have a phosphate group. So this 3' OH group, say this is the 3' OH which is attached to this gray portion. The 3' OH group over here now comes and it makes a attack, a nucleophilic attack onto the other strand. Once it makes that attack, ultimately we will have this thing coming out. This this region is now free. This has made an attack. So this segment is separate. We still have the signal sequences along with the other strand of DNA, right? So if you look here, let's just assume that one gene segment is out of the picture right now. It has already been cut off as a separate entity. So what we see here is something that looks like this. So this is a hairpin structure that is formed because of this attack. Now we will have endonucleases which can randomly cut at this hairpin. So it can cut this hairpin at this position, at any position at random, wherever it finds a consensus sequence. Endonuclease itself is another enzyme and it needs certain recognition sequence on the DNA to bind and act on it, right? So this endonuclease cleavage here, it is ultimately going to make a double strand break, right? So if you just imagine that this is now cut off, we have one gene segment with certain overhangs and we have another free gene segment. Now these overhangs and these points where we have certain cuts made, these are the sites where certain random nucleotide insertion happens. It can either be P addition or it can be N addition, right? So while we look into this image, we'll come to the details of it. Once these random insertions are made, ultimately we have these two green segments coming together and joining over with some additional nucleotides coming in between these joints, right? So this results into this ultimate stretch of DNA, which has a coding sequence and a signal sequence. And the ultimate joining of this is being mediated by certain double strand break repair enzymes. So some enzymes that are normally in use by the body for, say, whenever there is certain mutations that is repaired, or a break in a nick in the DNA backbone that has to be repaired. So whenever these things require any repair, we already have certain DNA repair machinery, certain enzymes that facilitate this process. The overall purpose of the RAG, the signal sequence, the recombinase enzyme is to mediate the cut and cleavages. Ultimately, the repair, the joining of the strand is being done by the usual repair machinery that comes into play whenever there is a cleavage for any other reason. So for recombination purpose, your enzymes that are majorly important is your recombinase enzymes, and the eventually whatever is the joining happening, it is being mediated by.
Recording in progress.
Okay, quickly give me a confirmation that everything is how it usually is right now. You can see me, hear me, and also see the screen.
Yes, ma'am. Everything is fine now.
Okay, okay, okay. I hope we do not face this again. I'm not at my usual place of work today. Fine. So now, now we can come to this diagram, right? So what we so far imagined is that we have a linear stretch of DNA, but possibility is such that when we have the orientation of our signal sequences of the gene segments, there is a possibility that we have our signal sequences in the same direction, right? So we have the coding, sorry, not the signal sequences, the coding frame, right? The orientation of the gene, which is determined by the promoter present. If we have the promoter present here, the reading frame is like this, right? And if we also have the promoter for the other gene segment, which now has to join, which is also here, so then the reading frame is falling in the same direction. So whenever we have two reading frame oriented in the same direction, what happens is known as a deletional joining, and it is a more common phenomenon. The other possibility, which however is less frequent, is that we have the reading frames in opposite direction. So if the promoter is present here, and the gene segment is going to be read in the opposite orientation, but we have, and that is why we also have the RSS sequences flanking opposite to each other instead of them facing towards each other. So that's something related to the orientation of the reading frame of your DNA, right? Which the RNA polymerase is now transcribing and all. So let's just take two possibilities simultaneously and we'll try to put everything that we have written here in the same in this diagram, right? Okay. So if we have these two frames together in the same orientation, what can happen is that this DNA, sorry, this region this region kinds of,
Are you writing?
yes.
Whatever I think that you are writing is not visible.
Yeah, 1 second. Is this now better? And my connection is lagging, I think. Okay, is this better now? Can you now see the frame? Whatever I'm doing. Is this okay now?
Yes, ma'am.
Just let me know if there's anything that goes wrong. Okay. Okay. So if we have these two things in the same orientation, what happens that we read that there's certain something called as the turn sequences, right? So there will be a looping, right? Because we have certain turns coming in, and the looping here is shown in this manner. So whenever we have this looping, because we have the rag sequence, the rag which is binding here and say here, because it has recognized the RSS sequences, it is trying to bend them and bring the two strands uh, in a loop in order to bring these two regions, which now has to be joined in a closer proximity, right? Is it still lagging? This is better. Okay. So now we have the first cleavage, the happening over here and over here, right? Ultimately, this 3 prime, as we were saying, is going to join. So we have a free end here and another free end here. This free end is going to join here in a hairpin-like structure that you see here. And similarly, everything same is happening in this parallel side as well. The only difference, if you see, is that the looping here is in this manner, and over here the DNA is bent in a in a single turn to bring these two things together. The rest, the formation or the cleavage or the formation of the hairpin structure, everything is the same. Now, when we have this attack of 3 prime OH group on the opposite strands 5 prime, so this is the attack that they have shown over here, right? And the another attack happening with this other gene segment. In overall, what you see here is that we have two hairpin loops forming. I hope what I am trying to convey is clear. Now, if you see further, let's just zoom this for. So this hairpin loop is now going to be subjected to another round of cleavage. But when this cleavage is going to happen, you will have certain overhangs, because if one thing is cleaved here, we have some stretch of DNA here, some stretch of DNA here, and any sort of cleavage is going to create a double strand break. And with this break, in an attempt to say fill the gap, we have some random nucleotide additions happening. There are same thing is also happening here. So this cleavage happened here, and we have a random nucleotide addition that is happening again. The only difference now, if you see, is when this cleavage has been made, we have one signal sequence and the other signal sequence along with this entire intervening DNA region. They are all together. But in this side where we have insertional deletion, insertional recombination happening, because the cleavage that has been made here is now completely separated off, and along with this coding region, we have the signal sequence from the other gene segment attached to it. It is all about the orientation, the way these two sites have been brought together. The ultimate result is, when you see here and here, we have a coding joint form between the V and J region, but this entire thing, the signal joint, is completely excised off in a separate loop. But here we have the signal joint associated along with this coding joint form, because this thing that we see here, we have the cleavage happening in this manner that doesn't take entirely excise of the signal joints, right? So that's how we have, if this turn is ultimately resolved and we again have the DNA which is now linear in structure, somewhere upstream or it might even be downstream, we have the signal joint which is maintained some base pair apart from the coding joint which has been formed. Right? So the only thing that we have for now not touched upon in detail, we will come to it towards the end of the class, is why this nucleotide addition is happening. See, we discussed that when we talk about antibody, there's a lot of diversity that is there. One way of accomplishing that diversity is, we saw that we have a multi-gene organization of the immunoglobulin genes, which means different gene segments are actually brought together by these recombination mechanism, which is going to create multiple possible combinations, because any segment can join to any J segment or any D segment. So we will have different combinations that is now coming to be possible. That is one mechanism, and that is what we are reading about right now. But we have seven such mechanisms in total, one of two of which is this p nucleotide addition is one of it, which is possible in both heavy as well as in light chain. So what happens if you look here, if you look at the sequences, this heptamer sequence that we talked about, it's a palindrome. What is a palindrome? That if you read it from this frame or from this frame, it reads the same thing. So the beauty of the heptamer sequence is that it's a palindromic sequence, and this nonamer sequence is a AT-rich sequence. So these things are actually recognized by the endonucleases that is going to cleave these hairpins, right? So what it does that whenever, so I had, so yeah, the cleavage is going to happen here, here, and these hairpins that you see. Now, why is it restricted to these region? Because of the signal sequences, the downstream sequence, the upstream sequence that serves as recognition sites. We then have the addition of P nucleotides on this region, which this is the J region here, and we have P nucleotides being added to the region flanking it, and we can also have some P nucleotide addition towards this others region. So when we have the ultimate coding joint form, what you essentially now have is the V and J that we have been drawing so far. But in addition to it, some base pairs have changed because of P nucleotide addition. This is possible for your both heavy and light chain. However, in the heavy chain alone, there is another new this TTD enzyme. This comes into role, and we have another possibility of N nucleotide addition. So N means any nucleotide can come and add to this region, and this is only happening in heavy chain, right? If you look here, so this we read about, so this diagram is helpful if you have to visualize that why we have a deletional joint or an insertional joint forming. But this is more helpful in understanding that what is the sequence, where is the cut being made, what hairpin and everything. So we'll just, this is the same thing that we have discussed in the previous diagram. We are just trying to see it in a different perspective. So this rag 1, rag 2 happening, what we were looking there is there's some looping happening and everything. So people have not drawn, the authors have not drawn that looping here. They have simply shown that because of this rag 1 and rag 2, one of the nucleotide DNA strand is now cleaved, and we have a free 3' OH and a phosphate. Similarly, so this is your V segment and J segment. We are talking essentially about the light chain. So they do not have any D segment in between. Right? But if there's a D segment, it's the same thing happening. So another nick is present over here. So your rag 1 and rag 2 nick on one strand. Now this 3' of free 3' OH is going to attack here. This is forming a coding end. And this other side where we have a double strand break is forming a signal end. Now these, if you see, are blunt ends, and over here we have a hairpin that has formed. These hairpins are further cleaved off, right? And because of which we have some overhangs left. So the AT-rich segment that we saw was corresponding to the nonamer sequence, and we saw that the heptamer sequence is a palindrome, which some way was a CG-rich site. So now we can have random nucleotide addition. We can also have, because the cleavage can be anywhere, so instead of the cleavage happening in somewhere near T, we could also have the cleavage happen here. So some nucleotides could be deleted, and then there is some random insertion of nucleotides. So all sort of addition and trimming of nucleotides can happen. We can also have alongside of this palindromic heptamer sequence, there can be some random nucleotides getting added on one strand of the DNA. So whenever we have one strand of the DNA having some randomly inserted nucleotide, what happens now, since we will have the normal filling using the normal repair machinery, what happens, we have complementary nucleotides getting added to the other strand as well. Ultimately, then once this gap has been filled, what we are left with ultimately is again a double strand break, because this is like having no overhang, this is also not having any overhang. We then have these two segments joined together using this mechanism of non-homologous end joining. So this is again a mechanism which is there in your usual repair machinery. Right? So this is what happens. This is the mechanism for joining of the different DNA strands in the process of recombination. I hope so far this is clear. Is there anything that you want to ask? Then you can ask me right now. This is now followed by that we discussed about class switching. Also discussed this diagram. There is another region. So we read about the VDJ regions. We have read about the RSS sequences. Now we will come to what there is something called a switch region, which is specific to the constant region. So VDJ, all this recombination, everything is happening in the variable region of your light as well as the heavy chain, respectively. But the heavy chain constant region can also be having some sort of recombinations happening, some RNA splicing happening. So they have the same VDJ commitment, but the constant region is different. And as we have looked about it previously, that the constant region of your antibody determines the biological effector response, right? So different constant region genes can be joined to this maintained combination of VDJ. So the antigenic specificity remains the same, but the class of antibody can become different. Okay, so if there is nothing to ask right now, let's just move ahead. So class switching happens. We have talked about it in different contexts. Where is it happening? It is happening inside of the germinal centers. So as I mentioned in the last class, all the previous 3, 4 classes have to be, you have to really remember everything that we have discussed in the last class and then try and put things in context, right? So we have discussed when we were discussing the development of B cell that within the germinal center, we have this class switching happening where inside the dark zone, right? Or inside the light zone. We'll see where it is happening, right? We at the end of this discussion, we will have a summary discussion of whatever is happening with the B cell and the receptors of it. What determines this switch is whatever is the cytokine that is present. This also we have talked about. How is this happening? The mechanism part of it. Again, we have certain flanking sequences called the switch region, which is a fixed number of base pairs upstream of all the constant region of the heavy chain gene segment. If you look about the arrangement of, so we have also discussed this previously, that for the heavy chain, we have different constant regions arranged in say upstream or downstream with respect to each other. And since we were talking about it, that why C mu, so why we have IgM and IgD always expressed together is because they are in very close vicinity, and the chances of both of these segments to get spliced off separately is not very likely because it is in very close proximity. And what is this close proximity? Why, if there is an additional distance, why is the chance of say IgM and IgG not expressed together? Because in this stretch of DNA, we also have something known as the switch region. So switch regions are short stretches of these repeats, right? So you do not have to really remember the sequences, but remember that the sequence of the nucleotide is actually serving a purpose. Different enzymes that mediate this process. So in this case, we have the enzyme switch recombinase that is recognizing these switch regions. So once we have the switch recombinase binding this switch region, again, there can be some sort of looping that happens, and this loop will ultimately be excised off. What does that mean? So initially, so if you look here, we have the VDJ recombination, which has already happened, and nothing is going to change anything with this thing, right? So the antigenic specificity is going to be maintained. The variable region is now fixed. The only thing that can now change the only thing that can now change is that which isotype. So which isotype determined by the switch region that is coming into play, the isotype can be changing. So if we have the S mu, so we have different constant region corresponding gene segments arranged in a linear fashion, and upstream of each constant region, we have a switch region. Only C del, if you look here, is not having any switch region of its own. So the switch region upstream of C mu and C del, then we have this followed by a switch region corresponding to the IG3 type, right? So IG also has subtypes that we talked about. So IgG of the IgG3 is there, and we have a switch region corresponding to that. The likelihood of this switch region joining with any of this switch region is equally possible. What determines it is what cytokine is present in the periphery of this developing lymphocyte, of this developing B cell, right? Let's just take an example where we have the switch region of the IgM, so the C mu region, which is ultimately going to code for IgM or IgD. If the switch region of this particular segment going to recombine with the switch region of, say, Ig1 segment, what happens? Whatever is now in between, so the C mu, the C del, switch region of the gamma 3, as well as the gamma 3 segment itself, all of this is now looped out, right? So there's going to be cut here and here, and this fragment is going to be lost, and this loss is forever, right? So you can never have the expression. So the switch has now happened. This is going to be followed by all different switchings again. So there is a switch here, then it can be another DNA looping and recombination, and ultimately we can have any of the expressing combination. So you can have this combination, this can again form a switch here. The only thing that is no longer possible is that whatever is excised off is lost permanently. So once you have the switch from IgM to, in this example, say IgG1, it is not possible for the switching to now happen to back to IgD or IgG3 because this has already been lost. We still have the possibility of it switching over to IgG2 because the gene segment is still present. But if there happens a switch, we can never have it revert back to the IgG1 type. It's all about how the recombination is happening in the constant region of the heavy chain that determines your isotype, and what signals that which switch should happen is the cytokines which we have. So this is something that you need to memorize, that which cytokine triggers differentiation of your proliferating B cells into which plasma cells. But all these are going to trigger different transcription factor activation, and based on this, you can have any switching possible. Okay, so one thing that we discussed today is the mechanism of VDJ joining. Other thing is the mechanism of class switching. And now we are going to come to another. So today's class is a wrap up for the B cell development that we have been talking so far of, right? Every mechanism that is behind whatever we have so far discussed for the B cell receptor. This is what we are discussing in today's class. So VDJ recombination we have discussed. We have discussed the isotype switching mechanism, the other mechanism that the other principle that actually underlies that how we can have the same antibody in secretory form or in the same antibody in membrane bound form. So for that, there is this process of differential RNA processing that determines that whether the antibody molecule that is encoded will be a membrane bound form or it will be in its secreted form. What changes? So if you so let's just imagine a cell, right? So how is a cell membrane? So this is the outer cell membrane, right? Outer membrane, and this is the inner membrane. Inside of the cell, we have the cytoplasm. So essentially, it's a water-based content inside this membrane-bound structure. So if you need something to be membrane, to be membrane-bound, it needs to have a transmembrane domain. And transmembrane, if you remember, if you have had a chance to read about cell biology, transmembrane domain is also a hydrophobic region because there is no water inside this region. So if this is a hydrophobic region, we will have a lot of hydrophobic amino acids that needs to be present here and should be there. Why so? Because it has to be present in a hydrophobic bilayer, lipid bilayer. But then it also has a small tail, right? So this is a C terminal which is inside the cytoplasm. And this tail should be hydrophilic now. Why so? Because it is there inside the cytoplasm, which is essentially a lot of water make. And that's why if something has to stay inside, it has to be hydrophilic. And again, if there is something that has to stay on the periphery, on outside of the cell, anything that is decorated on the surface of the cell, where do we have the cells? It's there in the fluids of the body. So again, it has to be hydrophilic. So we have a hydrophobic domain, a hydrophilic region, and again a hydrophilic region. Now, if an antibody has to be present inside the cell in its soluble form, in its secreted form, the gene segment, or sorry, not the gene segment, the RNA, the final RNA transcript that is formed has undergone a differential RNA processing, because of which it has lost its hydrophobic domain. So if it has lost its hydrophobic domain, it is never going to get a chance to get embedded into the cell, into this through, and it will never be able to cross the cell membrane, and thus it is never going to be in a membrane bound form. Is this clear? Okay, so and if we have a other possible RNA processing that has happened, which retains the all so red what you see here is all the hydrophobic amino acid, and this forms your hydrophobic domain. And if there is ultimately if the final RNA transcript has this hydrophobic domain, the protein that is going to get synthesized is going to have a hydrophobic domain which facilitates its crossing of the cell membrane, and it remains bound or decorated as a membrane bound antibody. So again, the different ways the gene segment arrangement and everything is there that has been shown here. We will not get into a lot of detail with this. You just have to remember the principle behind it. I am not sure if this has been discussed in detail in the lectures. But how is this happening? So if you look at the antibody structure, we said that we have one variable region, a constant, so CD1, 2, 3, all those things you were saying. So what is there? We have a constant region 1, 2, 3, and 4. If you're talking about IgM, if you're talking about IgG, we only have three such segments. So all that background you must remember. Now, if you look here, so what is being coded by the so CH1 region, say of the antibody IgM, corresponding transcript is present here. Here, the C for CD2 and CD, not CD, the region, the CH1, CH2 domains that we have discussed in the antibody structure. The corresponding transcript is here. After we have the the transcript that is ultimately going to be encoding the CH4 domain of IgM antibody, we can have a polyadenylation site present just downstream of this mu4. So mu4, I am not going to repeat this. So mu4 is coding for the CH4 region of IgM, right? We have a polyadenylation site right behind this. The other possibility is, so there's some site called as M1, M2 sequences, and we can have another polyadenylation site here. So this M1, M2 sequences is ultimately going to be your transmembrane domain. So the amino acids that's going to encode for this M1 and M2 stretch is going to be encoding hydrophobic amino acid residue. Now, while this primary transcript is being transcribed or is being translated, you can have the ribosomes adding a poly A. So this poly A site could be picked up while the processing is happening. And ultimately, if this is the transcript produced, it is going to be the ribosome that is going to translate the protein is going to have this protein which will be secreted up. Why so? Because there is no hydrophobic domain here which got translated. Why so? Because the RNA transcript in this case has been processed in a way where this poly A site has been selected for and rest of the regions has been lost. If you keep seeing the fact that nothing is changing with the VDJ combination which has already got rearranged. The other possibility is that we do not, so this poly A site is rejected for, and ultimately the splicing that has happened that has incorporated your M1 and M2 in the processed transcript, and ultimately the translation product is going to be encoding for a membrane bound IgM molecule because there is a transmembrane hydrophobic domain present in the translated treaty. Okay, so uh, that's another way by which we have the same gene. So if you see, the idea is to appreciate that this one chromosome, which has so many genes, so many flanking regions, different sequences, all of them have meaning in different contexts and in different mechanisms, right? So we often see the fact that we have this encoding region. This is the gene segment, but so many flanking regions are important that because of which we have different RNA transcripts coming from the same primary transcript because certain sequences were there and which mediated regulated excision or RNA splicing. So if you get into the details and you try to appreciate that who planned on to all this diversity, it's a very beautiful system. Okay, so two mechanisms discussed, isotype switching and how secreted and membrane-bound antibody is determined. Then comes the phenomena of allelic exclusion. So allelic exclusion, let's just understand what is allelic exclusion. So for all cells in our body, we have one set of chromosome coming from the maternal side and the other set of chromosome coming from a paternal side. and we are all diploid organisms, right? So any gene which is present on either of these chromosomes is going to have a chance to get expressed. But what happens in lymphocytes is that either paternal or maternal gene segments are expressed. So all this VDJ recombination, all the other kinds of recombination that we have discussed so far, look here. So when we have a progenitor cell or a hematopoietic stem cell, all the so this is for the kappa chain, the lambda chain, and the heavy chain. So different colors encode different sequences. So this is the maternal copy that this cell has received, and this is the paternal combination the cell has received. So ultimately, in the diploid organism, we will have a maternal chromosome as well as a a maternal chromosome gene copy and a paternal gene copy, both of them encoding for a kappa chain. Similarly for lambda chain and similarly for heavy chain. But when we have the rearrangement happening, it is not that we will have both of them having a equal chance of getting expressed. Either the maternal kappa is going to get expressed or the paternal kappa is going to get expressed. And that's true for lambda as well as the heavy chains. What is the purpose of this? So this phenomena where only one allele is expressed by the cell, despite of it having two alleles, this phenomena is known as allelic exclusion. So the expressed, rearranged heavy and light chains, they come from only one chromosome, either paternal or maternal. This phenomena is known as allelic exclusion. What is the purpose of? The purpose of it is to ensure that we only have B cells which are monospecific, that it can only bind to antigen of a particular type. Imagine a situation where the maternal chromosome kappa, the maternal gene for kappa is getting expressed, is undergoing independent rearrangement, and the paternal gene for kappa is also expressed and is independently undergoing gene rearrangement. So ultimately, we will have two kappa chains, both having some different combination, because recombination is a random process. The chances of the recombination resulting into the exact copy is firstly it's negligible in case of its possibility, and then also consider the fact that they have come from two different individuals which might be having an entirely different genetic makeup possible, right? So them landing up on the same possible combination is impossible. So if it's impossible, what will happen that you will have the B cell having antibody molecule which can different antibody molecule which can recognize different antigen. But the body wants that the B cell, each B cell should be specific to one kind of antigen only. Why it is so, the reason behind this is not very well understood, but we do want antibodies, we do want the B cell to be monospecific. And that is why one of the allele is always silenced. Now, I'll also introduce the fact that once we have the recombination happening, the VDJ segments joining, coming together, this recombination not necessarily will always form a productive, because you saw that there is some random insertions are also happening. If there is random insertions happening, it remains a possibility that sometimes you will have certain stop codons that have come together in successive trinucleotides, right? So if we can have different trinucleotides which are now encoding for so not encoding, so it's transcribing into a stop codon signal. So your ribosome is going to fall off and your translated product is not going to be functional because a stop codon has come in between, right? So such sort of arrangement, such sort of translation or rearrangement where we do not have a functional product forming, it is known as non-productive rearrangement. We will come to this also. We will have proper diagrams and everything to discuss this. But let's just know that some rearrangements can bring about productive recombination, and we can have some rearrangements bringing about non-productive recombination. So whenever we have a non-productive recombination, so in allelic exclusion, or let's put it in context of B cell development. First, it's your heavy chain that rearranges. So in the heavy chain, you can either have the maternal heavy chain copy that gets a chance for undergoing VDJ recombination, and let's say it was productive. So the cell, the product of that comes out from this productive recombination is going to signal the rags to fall off, and your heavy chain is not going to give about it's going to stop the process of recombination. It will not enter another round of recombination. Then we will have a signal to initiate the recombination for kappa chain. When a kappa chain recombination has to happen, both maternal and paternal alleles stand a chance, but one of the alleles is randomly selected, and say the paternal copy is selected and it undergoes rearrangement. The kappa chain that forms out of kappa rearrangement, say it is a non-productive rearrangement and we do not have a functional kappa that got coded. It is first given a chance to enter the process of recombination once again, because so this is a negative inhibition that is happening, that whenever we have a productive functional transcript forming, this product is going to inhibit the chances of another round of recombination. If we have the paternal kappa coming in, the recombination happens and it is forming a non-productive recombination, non-functional transcript. So we do not have a product to signal for stopping the recombination. Hence, it is given another chance to undergo the process of recombination. If it again results into a non-productive recombination product, now your maternal allele is switched on and it is now given a chance to enter recombination. So only when your one of the copies of your from your parents twice results into non-productive recombination product, your other allele from the other parent gets a chance to enter the cycle for recombination. So heavy chain. So let's now say that the maternal allele for kappa resulted into entered recombination and resulted into a productive combination. So we already have a heavy chain that has come. We again have a light chain, a kappa chain that has come, and we will have them two coming together. Disulfide bonds would form, and we will have a functional antibody molecule decorated on the surface of the mature B cell. Only in case of both the allele of kappa chain failing twice to give a productive rearrangement, then only the lambda chain, one of the alleles again for lambda chain gets a chance to enter recombination. So that is the sequence. One allele of heavy chain gets in, gives a productive recombination, the kappa chain recombination starts. So this is exactly what is demonstrated in the diagram here, right? So this is the progenitor cell. Heavy chain recombination. First is the D and J combination that happens. And then we have the VDJ combination that is going to form a functional heavy chain. If this combination is productive, will further have the V of kappa and J of kappa coming together and a IgG molecule is going to form. This if the line is entirely giving out productive alleles. But if after this recombination, a non-productive allele forms, we will again have a rearrangement that is going to be initiated. If we still have a non-productive combination, because now both the alleles is exhausted, the cell is triggered for death. But let's just assume that one non-productive recombination followed by another productive recombination gives me a chance for this cell to now enter recombination for its kappa regions, right? So kappa region again. This line evolved into a productive allele, but if this is non-productive, again another chance of recombination. If it now gives a productive combination, it is going to get selected, and we have a fully functional antibody molecule on this cell. If this is non-productive, so far we were having the kappa chain that was getting a chance, the kappa genes were getting a chance to undergo recombination, but now we have the lambda that is coming up. So if here we have the lambda chain which got rearranged in productively, it can get synthesized and the cells can get in further. If this is non-productive, a second chance is given for rearrangement. If we then have a productive O, the cell is selected, and otherwise the cell is ultimately triggered for death. If it has failed and exhausted its chances of recombining into a productive allele for all the types, it is going to be triggered for cell death. Is this clear so far?
Ma'am, here the 1 and 2 are representing the paternal and maternal.
Yes, yes, yes. Over here, it's talking about the paternal and maternal genes. It's not about the two chances given at recombining that I was saying. They're only talking in terms of alleles.
Ma'am, here in the case of antibody, there is no case of dominancy which comes in to context, right?
No, no,
It
no,
is
no.
just the
It's
expression
a
of
random chance.
every time the antibody formation is taking place, so every time this allele exclusion is taking place, or
Yes.
it is the fixed that only maternal or the paternal will going to express and form the antibody.
No. So over there, see, during the process of clonal selection, we have to have the variable region fixed. And this entire allelic exclusion, let's just try and understand it logically. If we do not have the same maternal and paternal combination coming up, how will we have the same antigen specificity, right? So, in clonal selection, to have the same antigenic specificity, it is important that we always have the same allele, either maternal or paternal, coming together and forming the antibody molecule. So, let's say if this particular cell is clonally selected, it now has to undergo clonal proliferation. The heavy chain recombination has to be from the allele 1, and the light chain also should be kappa from allele 1. But if, say, this chain was selected for clonal proliferation, if this antibody molecule got selected, so if it has come from this line where the allele 2 gave a productive recombination, and whatever, if you can just trace it, we have allele 2 and then allele 1 of the lambda that gave out this particular combination. This has to be the same.
Okay, ma'am.
Okay. Okay, so this I think so this is exactly what I was saying so far, that if we have in phase gene segments joining such that we have a codon reading frame, we have a functional heavy or light chain forming. But if the phase it is out phase, so out phase is that it shifts such that the amino acid we do not have the particular codon coding for any amino acids, or the amino acids that have now formed because of the shift, it is not forming a functional protein of a functional heavy or light chain. So that is known as out of phase rearrangement, and this is going to form a non-functional immunoglobulin heavy or light chain. So this is the basis for productive and non-productive gene rearrangement. This is what we were seeing here. So the purpose of having two alleles is clear, because if there is non-productive rearrangement, we still have another allele that can be switched on. And also the purpose for allelic exclusion is clear, is because we want all our cells to be monospecific. Right? Okay. Now we will get into the further understanding of the fact that why there is antibody diversity. We have pretty much discussed the first and the second phenomena where we already understand that multiple genes actually have to come together to form to form the respective chains of your antibody molecule, and they can come together in different combinations of VDJ joining. And how many possibilities does this give rise to? So it might come up as a numerical. We understand the mechanisms, we have discussed the mechanisms, but just if there is any numerical that comes on the basis of it. So if someone says that this particular organism, this particular genus has 51 V regions, 27 D regions, and say 6 J regions, right? So, how many possible combinations of heavy and light chains or of the variable region of heavy chain can be formed? To do nothing, you have to just multiply all these numbers. So, the number of combinatorial VDJ and VJ joinings, the possible number of combinations is this many, and the possible combinatorial association of heavy and light chain. So, try and understand this, that we have this many number of heavy chains that can form by the joining of by the possible joining of either of these many numbers of B, D, and J segments. Similarly, if it is a kappa chain that comes in, these many different types of kappa chains can form, and these many types of lambda chains can form, and the number of types of antibody now that can form. So we can have these many numbers of different heavy chain molecules joining to this many number of kappa chain, and also we can have these many numbers joining to these many numbers of lambda chain, because it's equally likely. So we do say, because we saw that lambda gets the last chance to come and recombine, but if we are counting the number of possible combinations, ultimately we just multiply all of them together, because these many numbers are possible. So how many V, J, and D and all these regions for all the different chains? It's fixed, and it for every species. So for humans, this is the number that is known to us for all the different chains. So this is how ultimately we can have these many types of antibodies that can be formed by combinatorial joining of V, D, and J segments, and these are the numbers that has been mentioned here for mouse. So roughly if you see, it's somewhat the same. It's just for humans we have further more diversity by certain decimal points, but when it is multiplied to 10 raised to the power minus 6, that's also a very significant difference. The other, so if you remember from the discussions of our very initial class, we said that the order of diversity is in this order for antibody molecules. But by combinatorial diversification, we are only achieving some diversity in the order of 10 to the power 6, right? So there are, one thing that is clear from this is there must be some other factors that is also leading to enhancement of the diversity. This exactly is all the factors that is, so not this, because this is association part we have already accounted for. So these four mechanisms are almost increasing your, almost diversifying the possible combination by another 10,000 times, right? So we will come to each of it one by one. Okay. So if you just try, if you start feeling lost, you can take this slow. We still have a couple of more minutes. I hope this is okay. I hope the speed is okay with you.
The speed is already okay, but I'm trying to manage the things in my mind that how can I adjust all these things.
So after today's class, whatever we have discussed so far for B cells, you will have this entire notes available with you. Do not worry, we just have three more mechanisms to cover up. And another thing that I'm not very sure of is in how much detail these things have been discussed in the recorded lectures. The idea is to also impart you additional information through these live sessions, because what is there in the lectures is already available to all of you. So try going through the written content for all these class that you get today. And we are meeting on Tuesday again for getting into the T cells now. Once we have done the T cells also, we can try and take one class where you have all gone through all the written notes and whatever. If you're having any problem in visualizing things, we can try and simplify like a backbone that we try to do it the last class, and we can also try adding all these details to this one backbone. So that whenever you have this one picture, you have some sort of recollection for everything that is happening for B cell and for T cell. So we'll try to do that because I do understand that this is a little too much in the first go. But once you start reading it from the books, so books are very helpful because they have even more information than this. So when you try remember, read more than what you actually have to remember, it is quite some information that stays with you. But if you just read this much, it's a chance that you will again miss out another 10-20% from whatever information you already have. So read more so that ultimately what you retain is this what you have in the written form in form of notes. And my job is to give you easier, simpler visualization of the concepts. So once we are done with everything, we will try and arrange one class where we summarize everything in 1 hour's time span. Maybe that can help. Okay.
Yeah, sure,
So
ma'am. Ma'am, this the remaining three, four concepts will get over till 8.
yeah. Because these are all little things. So these are things. So it's just two more things there, and that's it. The selection part we have already done, and this is a summary diagram that we have already discussed. And I thought that we can keep it at the end of the class if we have some time, and we'll try to put things in context. Let's see if we can do it today. Otherwise, in the following class, whenever possible.
No, ma'am, I just asking for the sake of my asking doubts, so that you can continue with
Oh,
the.
I'll just finish this today because and then we can stop the recording and then we can have your doubt session. Not a problem because you are the only one who still hangs on. So not bad. Okay. So let me just finish it so that the recording is done in the in time, and anybody who is following through the recorded lectures can benefit from it, and then we can take on the doubts that you have, right? Okay. So the third part that is enhancing that is further enhancing the diversity is the junctional flexibility. What is happening in junctional flexibility is when we have the signal joint. So remember, when the joints are happening, we have a coding joint which is being formed by the coding gene segments, which is either the V segment, J segment, or D segment. And we have another possibility of joining of the signal joints together. Signal joints in most cases, because deletional recombination is more uh, frequently found. And in deletional recombination, the signal sequences are joined together and spliced off, right? So we are only left with the coding joints there. But we also have the possibility of insertional recombination where the signal joints are retained. Even if they are retained, the joining is more precise, which means that there is no gap or anything that has formed in between, or wherein certain nucleotides or something could get added. So, whatever is the expected sequence, we get the precise joining for your signal joints. But when people try to sequence the, how do they know that it's the same which was initially there, is after a final process transcript is formed, people have tried to sequence this transcript. So they found that the joining here is precise, but this joining of the coding joins, this in some way was imprecise. So imprecise, which means that see, if primary transcript was having ATGC sequence, people found a AGGC sequence. So maybe one or two, something was added, something was missing. So this is a imprecise joining. So the productive combination that people found, they saw that there is some alternate amino acids that are getting encoded rather than what is expected from the sequence of your primary transcript. And most of these uh, alternate amino acids, I'm in class, I'll call you. So all the variability that is coming up because of certain variable or alternate amino acids coming up, most of this variability was confined to the CD3 region. So if you remember again from the structure of antibody, in the variable region also we have certain hypervariable regions, right? What is why are those regions hypervariable? Those are hypervariable because those are actually the regions of coding joints where we have, so if we coming from this V, D and J segments, this thing is the coding joint that has formed between successive segments. And when this thing is assuming the structure of antibody molecule, so this entire thing is the variable region, right? So this is a heavy chain because there's also a D region. We say that we have some hypervariable regions. This is the coding segments are actually forming your hypervariable regions of the heavy chain and as well as for the light chain. Because when there is this joining happening, some sort of flexibility of the nucleotide that can be added is present there. And this is what we were discussing as P and N addition. So why this imprecise amino acid is coming up? The one thing is because of the joining, we can have different combinations coming. The second thing is purposefully different nucleotides are added or deleted. Let's just discuss about P addition first, wherein we talked about the hairpin. We looked at the hairpin that is formed, which is cleaved by endonuclease, which leaves a single-stranded overhang, right? So this picture over here, this is the hairpin that has formed. If there is a cleavage of this hairpin, we have some overhangs right here, right? Now, these overhangs are going to be filled in by repair enzymes, by complementary nucleotides. If you see here that there is a GA overhang that has formed, it has been filled up by a CT overhang. Now, since the action of endonuclease here is random, so it can it is also equally possible that instead of the endonuclease chopping off, leaving a GA and AT overhang, let's say it left a CT and CG overhang on the opposite strands. So the ultimately what would have been the combination coming up, because there is CT, we will have, oh, I just, so it's your subconscious that with your GA you are trying to write CT. Let's just say this is A and T where it was cut, right? So instead of GA, which was giving a CT in the daughter cell combination, we have AT possible because endonuclease can cut randomly anywhere. And corresponding to this AT, we can, instead of having CT, we can now have T and A. Did I write it correctly? या A pairs with T and T के साथ A pair करेगा. ठीक है. So instead of CT, it was very likely that we can have TA, and likewise, it is possible for us to have any nucleotide addition uh, by the random action of endonuclease. So this addition that is happening because of cleavage by endonuclease of the hairpin, this sort of addition is called as the P addition. The beauty of it is that the hairpin is always forming where we have the RSS sequences. That's why it's most likely that you will either have a AT region or a AG or AT region or a CG region, which is more susceptible to cleavage of endonuclease. But the overhang that is ultimately generated can have different nucleotide combination coming up. So the idea is that it is so random that you never know what comes up. And because you never know what comes up, the diversity is increasing multiple folds. The other thing that we talked about already is N addition, which is only happening in heavy chain. And I told you that we have another enzyme, TDT, terminal deoxynucleotidyl transferase. So, as the name suggests, it is going to randomly add some nucleotides on the terminal ends of your DNA fragments, right? This is only acted upon onto the heavy chain. So, this is something you have to remember, that N addition only happens in heavy chains. So it can add up to 15 nucleotides at the joining of either the DG joining. During the process of DG joining, also it can add up to 15 nucleotides. Or when we have the VH region joining to this combination of D and J, again, this joining can be acted upon by TDT, and we can again have up to 15 nucleotides added in this region. So, essentially, as a consequence of, so we say that after this joining, what do we say? That we have VH, DH, and JH. But if you try and incorporate the end nucleotides, you can also write it like we have VH and N nucleotides, some 1, 2, 3, up to 15 such bases possible, and maybe another, maybe 10 or even 15 such N nucleotides can be there before a J region is coming up, right? So because of this, the diversity index of the possible combination is greatly enhanced. So just
Excuse me, ma'am.
Mhm.
If I'm not wrong, then V, D, and J are making up the variable region of the antibody.
Yes.
So, ma'am, if this possible number of nucleotides are coming in between these sequences, then it won't affect the variable region.
It do affect the variable region, but if there is a non-productive arrangement that has now come up because of these random insertion, the cell is going to be triggered for cell death. So if you
So,
remember from
cell
previous
will
class,
be going to trigger off for cell death, or the antibody will be destroyed, or both will happen.
when if the antibody is not useful, the B cell is not useful. So the body is going to kill off. So either it is going to enter another round of recombination, as I was saying. If that is also not happening, the other allele is going to again given a chance. But during every process of recombination, such additions of nucleotides is happening. If as a consequence of nucleotide addition, we have a non-functional product forming, it is the cell that is going to die. Because if it cannot make useful antibodies, we don't want the cell. So that is why, if you remember from all our previous classes, 90% of the cells actually die because the combinations can either be reacting to self, they might not be having the ability. So if you remember in the germinal center, the follicular dendritic cells have certain antigens to it. हो सकता है कि वो सेल्फ एंटीजन से रिएक्ट कर रहा है तो भी डेड होगा. सबसे पहले तो if it is not able to form a functional product, it is going to die despite of all the possible number of recombination events that it could have entered with the all the alleles that it has, right? If it is the case that it is not forming any sort of useful rearrangement, it is going to die. After it has formed a useful rearrangement, if still it is not binding to the antigen with sufficient affinity, it is again going to die. So these cells are given a chance to
Ma'am, will you please wait for 2 minutes? I am having some random questions coming
yeah,
in
yeah,
mind. So before they disappear, I want to ask them before.
yeah. Please go ahead.
Ma'am, before the formation of the antibody to get functional, it must be come in contact with the antigen, right? And the
Mm-hmm.
contact with the antigen of that antibody is taking place inside the germinal center, right? That we have talked upon. But the formation of antibodies taking place inside the bone marrow. So from there, it is transferring to germinal layer. Am I right in this?
Yes, you're absolutely right.
And ma'am,
You first complete yours, huh?
yes, from there, the antibody molecule is coming in contact with the antigen, right? And now, if like we have shown right now in today's class, that certain antibodies, that you have told that certain antibodies can be formed, can be found fixed inside the transmembrane protein, and some found in the random, right? They are not bounded to the transmembrane protein. So in that case, if that antibody that is free, that is not bounded to the transmembrane protein, so in that case, how that cell will be triggered if that cell has already formed that antibody and release that inside the bloodstream? So in that case, how that cell will be leading to the death?
Okay, one by one. In the bone marrow, we have the VDJ recombination and everything, all the development is happening inside bone marrow for B cell, and for T cells, it's happening inside the thymus. So while in the bone marrow, it is all growing, developing. We have follicular dendritic cells, which present your developing B cell purposefully with self antigens, and that is where selection for anything reactive happens and the cell is triggered for death. We are assumed self, so even in a self reactive antibody, we have had productive recombination, but the combination was such that it is reacting to self. Only if a recombination productively has happened and it is able to recognize no self antigen, this cell gets a chance to leave the bone marrow and reach the peripheral lymphoid organs where they can form into germinal centers and all the possibilities, right? Inside the germinal center, once the antigen is trapped, so all bone marrow से जो cells निकले, this army is having huge sets of huge number of possible antigen that it can recognize to, and this diverse army reaches the lymphoid organ and it waits for an antigen encounter. Once the antigen comes in and maybe one of these cells identifies is able to bind this antigen because it has a VDJ combination that allows for binding to this particular antigen, this B cell gets a chance to undergo maturation and differentiation. The production of secreted antibody only happens when a antigen encounter has happened, right? So it is never that you will have non-product it's not like a non-productive combination has happened, the antibody has got secreted and it is complete waste, because a antibody in its secreted form only comes into picture when this antigen has bound a B cell with the antibody molecule. Now, if you remember the secreted form, the differentiation that happens, it comes next in picture. While now the genes that are getting transcribed, they will now be having the transcript is not going to have a hydrophobic domain anymore, because of this antigen which is already which has already been encountered. The VDJ region of this particular B cell is fixed. It's only that the transcript that now gets to that now forms will lack a hydrophobic region, and the antibody with the same specificity is going to come out in secretory form so that it can bind to the antigen on its own, which is not present on a cell, because the cell has got that instruction कि आप ये एंटीबॉडी का secreted form बनाओ ताकि वो ब्लड में कहीं और पहुंच सके. Antibody in secreted form is never formed if there is no antigen. Is
Okay,
is
ma'am.
it is it an answer to your question?
Yes, ma'am, I got that answer. And ma'am, the signal for the antibody production in secreted forms comes from the outside, right?
Yes, the cytokines.
Cytokines. So, ma'am, they are making changes in the plasma membrane of the B cell as well, because if we want to remove the hydrophobic portion, so then in that case, the signal must reach. No, ma'am, I got that concept.
So we have the VDJ, which is fixed, but we still have all the constant region, different isotypes possible. All the, say, the C mu, C del, CJ, everything is already present. It's just the VDJ combination that is fixed. The B cell first produces IgM and IgD with a particular make of VDJ. Upon antigen encounter, upon activation, everything that so accepting allelic exclusion that has already happened during the process of maturation, your even this VDJ recombination, N nucleotide addition, P addition, all of this has happened in your bone marrow while the cell was undergoing development, right? So your VDJ is fixed now. But when this cell is has to now undergo differentiation, there the switching, so you can have the switch regions activated, the switch recombinase activated in the presence of antigen, and your splicing to a different isotype of antibody is possible. So isotype switching is that's it's possible in presence of antigen. Apart from that, the next concept that we read, somatic hypermutation, that is also possible in a activated B cell. But this N addition, P addition, BDJ recombination, all of this, which locks the antigenic specificity of the antibody molecule, that has already happened in your developmental stage. This is not something that happens in response to antigen. In response to antigen, your class switching happens because of the switch recombinase that gets activated in presence of cytokines, and somatic hypermutation happens. Is this clear?
Okay, ma'am. So, ma'am, we can say that class switching and somatic hypermutation are producing the variability, or are providing the diversity to the antibody in presence, yeah, in response to the antigen,
In response to antigen.
and the
We
remaining.
can say that. See, somatic hypermutation also happens in exclusivity. Class switching happens only in presence of antigen. But when the B cell comes into the germinal center, It has a possibility based on, because inside the germinal center also you can have some cytokines, and I think there's also an enzyme that comes into picture, PCNA. PCNA is an enzyme that also comes into picture. So some sort of hypermutations also keeps happening. So hypermutation is something that keeps happening. If the mutation is going to produce higher affinity antibody, the cells are going to be selected for. If the hypermutation is going to produce the same affinity receptor, or it is going to decrease the affinity, the cells are going to be triggered for death. So that is something that is happening irrespective of antigens also, but in the peripheral lymphoid organs, not during the developmental course of the B cells. But your VDJ combination, the combinatorial joining of different H and different L chains, the allelic exclusion, that fixing that which allele has to be picked, your P and N addition, all of that, anything that locks the specificity of VDJ region is happening inside the bone marrow before the antigen encounter. Somatic hypermutation keeps happening. Your class switching happens only in presence of antigen.
Okay, ma'am, got that point.
So I understand that once we have one diagram that has everything, that is how it's a way to visualize everything. I'll try to do that. Okay. Okay, so N addition is clear. So ultimately, whatever the combinations that ultimately arise for is this VH, N, DH, N, and GH. And then we have the CH domains. Whatever CH comes in, that determines which antibody molecule has been formed. Okay, so this is a figure to explain N addition. So unlike, again, there is a cleavage of hairpin, because for any sort of addition to happen, we need a break, right? So whenever we have a break, then it is acted upon by TDT, which is randomly going to add any number of nucleotides. It could be any any nucleotide. So it can be ATG, any possible combination. So 15 such. So just imagine 15 nucleotides that can be added. So four nucleotides we have, 15 different, any of these four can be added in 15 number of times, multiple combinations. So this is one mechanism that tremendously adds on to the diversification of the antibody molecule, right? Ultimately, after n addition, we will have H chain, which can, so many different H chains coming up just because we have 15 nucleotides that can be added by this TDT enzyme. TDT enzyme is also a lymphocyte specific gene product like the RAG1 and RAG2. It is only present inside the lymphocytes, right? So these are the mechanism, and the last that comes in picture is somatic hypermutation. So, somatic hypermutation is the process wherein, again, we have random mutations happening. So far we saw that some nick is made, and in an attempt to refill the gap, certain nucleotides can be added or deleted that way, right? But over here, we have a single DNA, and this DNA can undergo a mutation. So we have so this helps to alter the antigenic specificity. This can alter the antigenic specificity of the antibody molecule that has formed, and also contribute to affinity maturation. The one possibility is the mutation happens such that the specificity of the antibody has changed. It can bind to a slightly different antigen. The other possibility is that now the binding affinity has increased. Say instead of a positive amino acid coming up, we have a negative amino acid now, which is better able to bind to a antigen which is having a lot of negative amino acids. So negative interaction in molecular biology we do not have. We have similar. So if a hydrophobic amino acid is going to prefer a hydrophobic amino acid, it's not opposite interaction there. So those sort of things can change because of somatic hypermutation, and the process that this process happens leading to affinity maturation. Where is it happening? It is happening inside the germinal centers, and the hypermutation is restricted. It doesn't happen on the entire gene fragment. It is only happening in the variable regions, particularly in the hypervariable regions or the CDRs, right? So CDRs are the hypervariable region. We have also read CDR in context of T cell receptor, but then that is a different thing. Here, CDR is the complementary determining regions or the hypervariable regions of antibody. If you look here, it has been shown that this is the VDJ segment, and the frequency of mutations is highest in this region. As we move towards the constant region, there is absolutely no mutation that is happening, right? So the somatic hypermutations are restricted to the variable region, and as we move away, as the distance increases from this VDJ region, the frequency of mutation decreases, and it is happening inside the germinal center in the centroblasts. So centroblasts, if you remember, we first have the so centroblast divides into centrocytes, which now have their uh, anti gen specificity fixed, right? But we will also see that the centrocytes, when they are entering into successive rounds of replication during the process of proliferation, they, by the process of hypermutation, are also trying to increase the affinity of binding of the antigen to the antibody molecule. So the mechanism involves substitution, insertion, deletion, like how we have been seeing throughout today's class. Substitution mechanism is more common, and deletion and insertional mutations is less common. If we look into the mechanism, we have two enzymes that play a role. The first is this enzyme called activation induced cytidine deaminase, or AID. So as the name suggests, cytidine deaminase, it works upon a cysteine residue and it deaminates it. So the structure of nucleotides, if you see cysteine and uracil residue, deamination of cysteine converts it into a uracil. Uracil is essentially not commonly there in the DNA, right? It is T that is present. Uracil is present in RNA molecules. But if we have the uracil present here, so deoxycytidine is converted to deoxyuridine. And now, whenever this replication is going to happen, a tendency to fit in with the uridine, deoxyuridine, is going to be for deoxyadenosine. So deoxyadenine. So if replication happens, okay. So let's just see. So we have a DNA backbone here. This is a C residue, and if it is so with C, we usually have the pairing of G. But now this C, because of the presence of action of 8, we have the C converting into a uracil residue, right? And a G residue. Now, when this particular strand, right, when this strand is going to enter in the process of replication, we will have these two strands separate out. When this strand enters replication, normal replication, what happens? The daughter cell, instead of, if it would have had C, it would have given a daughter cell which is CG combination again. But now, since it has U, the daughter cell that forms, instead of U, it has A, and ultimately, in subsequent generation, we will have a AT transition. So, such a mutation that changes your purines to pyrimidines. If I'm not wrong, you can check that thing. This sort of mutations where CT mutations is called a transition mutation. So replication will change this into a CT transition mutation. The other possibility is that, so these are all mechanisms that mediates this somatic hypermutation. Other possibility is that there is another enzyme called uracil DNA glycolase. The function of this enzyme is to remove uracil. So now if this, instead of entering replication, if this is further acted upon by, sorry, uracil DNA glycase, uracil DNA glycolase, then this changes, this removes, as the name suggests, it's going to remove the uracil, and what we have is just a G. So such sites where we have a nucleotide missing, this is known as a basic site, where we do not have any base. This A basic site is acted upon by a polymerase, which is a error prone polymerase. This error prone polymerase, which means that it tends to produce errors. So if it produces error, this is exactly what the body wants, because it is wanting to create mutations. So because of this error prone polymerase, this A basic site can be filled by either A, G, it can also be a C, a U, sorry, not U, can be a T, it can be a G, right? So it can fill it up with any, so this error prone polymerase can fill it up with any of the nucleotides. If it fills it up with any nucleotides, in the subsequent rounds, you will have a different combination possible, and that is how another set of mutation has been induced, right? What recruits this error-prone polymerase is this thing called PCNA, proliferating cell nuclear antigen, right? So this is, a second. PCNA, I am not sure that it's exactly an enzyme or if it's a transcription factor or something. I'll check that. It might be a transcription factor, a transcription because because it's these factors only that loads the polymerases. So it might be a transcription factor that is loading this polymerase, which is having a tendency of giving error-prone results. The other possibility, again, so this is the last thing that we discuss for today, is that it undergoes mismatch repair because a UG is not a correct match. So it can activate mismatch repair related enzyme. So by normal, how the mismatches are filled up. So initially in mismatch, we have a gap that is formed, and this is subsequently filled in by polymerases. But because of the presence of PCNA, the recruitment is again going to be of the error. Instead of the normal polymerase, we will again have the error-prone polymerase that gets recruited, and we will have different kinds of uh, the nucleotide bases that is incorporated, and in subsequent round we will have some totally different combination of nucleotide pair than what we can expect from the initial gene that was present, right? So these are the basic three mechanisms by which hypermutation is brought about inside a B cell receptor. we'll not get into selection for today. I can understand that this is all that where we should stop today, and we'll take this up into the next class. That's also a common thing that happens in B and T cells, so we can discuss it there. Okay. But somatic hypermutation, how is it brought about? Why is it brought about? Where is it happening? I hope all these aspects are clear. And these random mutations, uh, now because of these random mutation, the antigenic specificity can change. It might be that it is producing higher affinity. It might be the specificity has changed. It might be producing of a lesser affinity. So all these things have to be subjected to a round of selection. So this is the selection. There can be a positive selection where a cell is given a green signal to get activated. Antigen is there, get activated and do your job. If the selection is negative, the cell is triggered for either death. It might be given another chance by the process of receptor editing, where it is again allowed to. So, editing is exactly what it means. It's like a backspace and type some other word and move ahead. If that gives about increase in affinity, good. If that also fails, then there is a process of clonal energy. So those are mechanisms of negative selection. So we can either have a positive selection or a negative selection of all these centrocytes that have undergone somatic hypermutation. So we'll stop here for today. And if there is anything that is not clear, we will try and keep this as our framework, and we will put things in side of this diagram. So now, Shamika, you can ask me whatever questions you have. Take a moment, have a look, because multiple things is also mentioned in this diagram, as we have also previously discussed this, and then ask me that which all things are confusing you right now.
Ma'am, you have told that somatic hypermutation can also be done if there is no exposure or to the antigen has taken place, or I think that B cell activation has not been taken place. So
Mhm.
can you repeat that sentence?
So inside the bone marrow, we have the B cell development that is happening. So from the hematopoietic stem cell until the formation of a mature B cell that is antigenically committed. All these mature B cells get a chance to enter the lymphoid tissue, and it's the site for your antigen encounter. The hypersomatic hypermutation that happens, it can happen prior to antigen encounter, while the cell is just trying to divide inside your lymphoid organ. It can happen after the antigen activation, by which why so? Because the cell wants to bind to this antigen with higher affinity. So, somatic hypermutation can happen at all these stages, in presence of antigen as well as in absence of antigen. As a consequence of somatic hypermutation, the antigen specificity can increase, decrease, or even change. But then we have selection in place, which will only select for the higher affinity and the safe antibody molecules which are present on the cells. So non-self reactive and can bind to the antigen with higher affinity. If either of this is not the case, the cell can be taken for cell death. So if you look here, so here is the centroblast, which is a mature B cell but can undergo a lot of division. This centroblast in lymphoid organ inside the dark zone of germinal center can undergo somatic hypermutation as it is proliferating. So in every round of proliferation, there is DNA. Oh, okay. I had to also answer your query about what is germline DNA, right? So let's just discuss this and we'll come to that. So while the cell is dividing, it has a chance to every time uh, so every cell has constitutively every cell has the entire genome, right? This entire genome is actually the germline. Once a cell is dividing, it can divide into what it can produce. One second. When the cell is dividing, we can have the replication process occurs. Once the replication occurs, then it can, so in context of lymphocyte, it is, can also undergo a lot of rearrangement possibly. The rearrangements can, after the rearrangement is done, there can be somatic mutations, and there can, because of somatic mutation, process of affinity maturation can also be happening. But affinity to what? Affinity to this antigen interaction that is happening. So there is selection for high affinity centrocytes, and if the affinity is low, it is taken for cell death.
Okay, ma'am. So, ma'am, somatic hypermutation is not taking place in the bone marrow, right? It is taking place inside this when the B cells has been come from bone marrow to the lymphoid organs, right?
See,
At the time of
after,
proliferation, right? We can say.
you can say it is more meaningful when it is happening in the while it's proliferating, but inside the bone marrow, after the VDJ combinations have happened, the joining has happened. So there's no there's no clear line, because bone marrow is where your entire development as well as maturation is happening. So maturation is happening as a response to antigen. So antigen, it's not something that is, say, black and white, because for B cell, bone marrow is also the site for maturation. Antigen encounter is primarily happening inside your peripheral lymphoid organs. But we also have dendritic cells as a part of the bone marrow stromal network, right? Where we can also have some sort of antigens that is purposefully being presented to the B cell. So they can be self antigens, or they can also be foreign antigens. And the body wants to select for the B cell that can react to the foreign antigens only. So there also, for recognition purpose of antigen, you can have somatic hypermutation and some sort of those things happening there. So it's not like it will completely be absent there. But it is a phenomena, somatic hypermutation is a phenomena that is talked about in context of dark zone of germinal centers. Mutations, incorporation of nucleotides, everything of that sort is happening throughout the process of maturation. It's not a very strict line that just after it enters, it only then can undergo some mutation. Mutation is a underlying principle throughout the process of maturation, happening alongside this VDJ recombination that is happening.
Okay, ma'am. So I got that answer that dendritic cells are purposefully are present in the bone marrow, right? And from there, the maturation and all these things are happening, and all the exposure to the B cells are getting from there, from that dendritic cells, right?
Yes, yes, yes. So a
For the
purpose. Mhm.
and for the T cells as well, we are having the dendritic cells present in the thymus.
Yes, exactly.
Okay, ma'am. And ma'am, if certain changes or the mutation has been taken place in the antigen, then the corresponding antibody, which has already been formed inside our body, that will also undergo some sort of mutation, or it will be uh, then another antibody will be formed with respect to that antigen.
See, if the antigen has undergone mutation, which is very likely the case because the microbes are always trying to mutate and form something to escape our system, right? So, see, the idea is that we have B cells which can recognize to n number of diversity. So if the antigen has undergone certain sort of mutation and we have a new attack of, say, a different strain of a virus or a bacteria or something like that, there must be some other uh, soldier which can take care of this. The previous one, which was undergoing proliferation and all those things, clonal selection and everything, it might not very well take care of this new challenge. So body recruits the other one to take care of it, which was made particularly for that particular antigen. Now, while the body was making it, it did not know that it might encounter that antigen. But it's a matter of chance that body made it because it can make a huge lot of diversity and it recognized that antigen, and it was a useful diversification that was done. And in terms of if you are saying that, say, the antigen which was so like might be so that is one case where the entire organism has mutated into a different strain or something like that, and it is a fresh encounter. The other possibility is that your antigen is the same. It's just that it has modified a little, say like a one or two amino acids. So for taking care of that, these cells of your body again is also undergoing hypermutation. So whatever is going to take care of this particular changes in the antigen is it in the antigen, if it is capable, if the somatic hypermutations are good enough to bind to it, because this process is also producing different antibody molecules, right? Different cells with different antibody. If very slight changes is there in the present encounter, the somatic hypermutations can take care of it. But if it's like a completely different strain which has undergone, has accumulated multiple mutations together, you can count on your uh, diverse repertoire of B cells, and maybe some new soldier will take care of it.
Okay, ma'am. And ma'am, in the somatic hypermutation we have saw, so at the time of replication, there are certain enzymes also which are taking care of that proper DNA strands should get formed. So in that case, that enzyme will not be in much use, that we have shown that error prone, some, I'm not remembering the name of that enzyme.
Error
Some,
prone DNA polymerase.
yes, ma'am. So, if they are acting, then that DNA, then that enzymes which are taking care of that that proper sequencing or nucleotides arrangement should be present on the another strand of the DNA, so that won't get affected by the activity of this error-prone polymerase.
If it gets affected, the body is going to eliminate that cell. They do get affected. So that is what I am saying, that after hypermutation, it has to undergo selection, which was the next topic. So in the process of selection, body eliminates anything that is now less useful. So again, almost 90 to 95% of these cells, which are going to form as a consequence of it, is going to be eliminated.
Okay, ma'am. I'm thinking that most of my questions answers are, somewhat linked together.
Yes, yes. So that if you are feeling so, then you are understanding the things correctly. So immunology, that is exactly where we started from. that it's not one thing, it's multiple things, everything happening together, everything happening as a consequence of the other thing. We are trying to break them into different topics, but once we have covered everything, it is important for us to visualize everything as a common occurrence, everything happening simultaneously. So if you look here, while we initially started with this diagram to just understand that what is the different cell type that is coming up, you would see that this also combines a lot of names that we randomly just mentioned over there. So while the cell divisions are happening and one cell is changing to the other cell type, so centroblast से centrocytes बना, उससे plasma cells बना, plasmablast बना, while all of this is happening inside the nucleus of the cell, your expression pattern, recombination, all the hypermutation, this gene switching, everything is happening simultaneously, right? So of course, every question's answer is going to be related to the is going to give you a next question, or the answer to that question might answer some other question. So it is very interlinked.
Okay, ma'am. And ma'am, in today's class, you were explaining the first topic. So in that, you have taken the example of, so in that, what is the thin gray region? It is the simple double-stranded DNA that you were trying to explain it from the your
Earphones.
head.
Yeah.
Yes, ma'am.
Yeah. It's just the strand of DNA.
So, ma'am, that is not coding for the any particular protein. So it
No.
is a non-proteinaceous part. Matlab,
Yes.
we can say that it is not a gene,
Yes.
right?
So we have the coding joins and coding sequences, and as the name suggests, they are going to code for your variable segments of your light chain or the heavy chain. But the signal sequences, we also saw that the signal sequences can just get entirely spliced off, right? These signal sequences are never going to, if it is spliced off, the purpose of it is over. But if you remember that, so we also have something called this LUC here. This is a
Um,
leader. Mhm.
no, ma'am, you
So,
can continue.
so while we were also discussing the VDJ and everything, this sequence, this leader sequence, as I was saying, it's a signal for the entry of these heavy chain, light chain, variable regions to enter into the endoplasmic reticulum where the folding has to happen. So ultimately, when the immunoglobulin molecule is formed, this L chain, this L segment that you see everywhere, that is also completely taken off. So that also doesn't form a part of the ultimate protein, the immunoglobulin molecule that you see. So what you ultimately see and what was initially present is completely different. So the signal joint that was present so far, the entire purpose of the RSS sequences is to guide this process of recombination. It is not coding for anything. It is just serving as sites for binding of your enzymes, the RAG1, RAG2 molecules. So,
Signal sequences and RSS both means the same now.
yeah. It's RSS recombination signal sequences.
Okay, ma'am. So, ma'am, the deletion joining is ultimately leading to the formation of the, useful genes, right? And it is eliminating the non-useful part of the DNA that we have
Yes,
shown on your example. And it
yes.
is removing the RSS as well because its role got over. And ma'am, while explaining with your earphone, you are trying that to explain that it has cut it down the one gene, and now it has to act on, and now the another gene has to be broken off. So how that is being taking place?
See here, we saw that there is a, see this is what I was trying, that so this is the strand that I was we brought up, right? And we say that these two green things, maybe this is the V and the J segments, right? So this cut, if I have to cut here, so maybe this is the cut that I am talking about, right? And once I have this thing cut off, this can like lie around like this, but this 3 prime OH makes an attack at this joint, the RSS signal of the second gene segment, the V segment that comes up. So I like that's what I was saying, that I cannot, like, let's just assume that this J segment is already free, right? And this is the hairpin structure that you see. So so hairpin structure that they have made like this, right? Now this hairpin can be acted upon by endonucleases at different points, and then we have the nucleotide addition and everything possible.
So, ma'am, that
Ultimately, mhm,
so that the removed portion is getting to be joined in between this uh, cuts that has endonuclease has made.
yeah. So if the endonuclease, say if the endonuclease uh, cuts somewhere, say here, right? So what we are left off is this part, right? This much of this and the further VG region, let's say. So if it cuts here, according to this, we can have different base pair addition that is possibly happening. So because of that, we have all the p nucleotide addition and n nucleotide addition that is happening. So if you look here, see, this is more, I think, if I would have been in a position to tear this off, but this is exactly what is happening here, that we have one cut that happened here, right? And then we have p additions. Similarly, another cut that has, so here if you see, it's not just it's one strand that has been cut, right? So there it's not possible because it's just exactly one strand that I exactly have in hand. But of the two, we first have one cut, another cut, p additions, and ultimately this getting joined, and this region coming and joining into a coding joint, and these two joining together into a signal joint. So this becomes like a circular molecule, and it is eventually degraded off.
Ma'am, how that 3' phosphate group is making attack to this another gene? Because it is not an attacking reagent, we can say, or we can say that it is an attacking nucleophile.
It is a free 3 prime. So if you see why we have this DNA replication always happening 5 prime, 3 prime direction, because the 3 prime OH group, it is something that can make a nucleophilic attack. So that is the whole reason. So as soon as we have a free 3 prime OH, it has everything that it needs to make an attack on this strand now, and that is what eventually forms this hairpin loop.
Okay, ma'am, got that point.
Should we go over anything else?
Can you just scroll it up?
Here.
Yes, ma'am, on this page only.
Mm-hmm.
Ma'am, actually, I'm not getting your voice that what you have started with this, but I got that whole concept very clear. So can you just brief me out that what you have explained with this?
See, so once there are two possibility: transmembrane domain and secreted form. For any protein to be having a transmembrane domain, it must have certain hydrophobic amino acid residues, because the core of this transmembrane region is hydrophobic in nature. So if because of RNA processing, there are two poly A sites, right? If the RNA processing happens in a way that poly A additions happens here, we will not have these M1, M2 regions, which are the hydrophobic which ultimately gets translated as the hydrophobic domain of the protein. So if I have the polyadenylational occurring here, This is the thing that forms, and the RNA transcript is going to be for a secreted form of antibody. But if the RNA processing is happening such that the poly A is is going to be added after the M2 region, if this poly A site is picked up, then ultimately we have a transcript that has M1, M2, which is the transmembrane, which is going to get translated into the hydrophobic amino acid residues that eventually make up the transmembrane domain of your membrane-bound antibody.
Okay, ma'am. So, ma'am, from the secreted protein form only, they are getting embedded inside the plasma membrane. It is not like that the some structure are present already inside the plasma membrane and then the protein is getting attached to that portion, right?
No, yeah. Because see, so this is something from cell biology. While where is replication happening? It's happening in the nucleus. The process of transcription again happening inside the nucleus, and then we have the transcript that comes into the cytoplasm. The process of translation happens in cytoplasm. So the cell has produced the immunoglobulin molecule and everything inside the cytoplasm. But every protein that is being formed, it might remain intracellular. It might have to be a excreted protein, secreted protein, or it can be a transmembrane protein. So we have the endoplasmic reticulum that does this job, where the translated proteins are given certain tags. These tags are recognized by either the Golgi or the endoplasmic reticulum, and eventually they are transported to the region of cell where they are supposed to mediate its function. So similarly over here, your secreted form as well as your membrane bound form, both of them is completely translated inside the cytoplasm. But based on the folding that it can presume, so we have this leader sequence. This leader sequence actually guides it inside the endoplasmic reticulum. Next is the job of endoplasmic reticulum and Golgi apparatus to see that what is the which folding it can assume, and based on that it packs different proteins into different sorts of vesicles. If it is a transmembrane protein that has got packed, the vesicle fuses with the cell membrane in so if it's a secreted protein, so we have this vesicle wherein the secreted form is packed, and when this is going to fuse with the outer cell membrane, whatever is the content of the vesicle is going to come into the external of the cell in a secreted form. But if it has to be a transmembrane protein, while the import inside the endoplasmic reticulum is happening, the protein doesn't completely gets inside the lumen of the reticulum, right? Right from its entry, it partially enters the endoplasmic reticulum, and this hydrophobic region stays embedded inside the so endoplasmic reticulum is also a bilayer structure. So this is everything from cell biology. You have to, I'm not sure if you have read about it. So this bilayer structure, the hydrophobic region of your transmembrane proteins are captured from the very beginning, bound to the membrane of your bilayered endoplasmic reticulum. A small portion of this membrane is going to be pinched off, and eventually a vesicle is going to form where we have the transmembrane protein embedded on the membranes of the vesicle. So when now the vesicle fuses to the cell membrane, instead of this protein coming out, if it would have been packed inside the space of vesicle, the fusion of vesicle is going to secrete the protein out. But if the protein is present bound to the membrane of the vesicle, fusion of the vesicle to the cell membrane is going to express this transmembrane protein in the bound form on the cell membrane. This is endoplasmic trafficking and endoplasmic reticulum trafficking. This is the topic which you should read if you want to understand that, if you want to understand what I just said in a better manner.
Okay, ma'am. And ma'am, it is for sure that polyadenylated residues are going to be added up at the 3' end and the mm, and the Ma'am, this is the correct statement I want to sure.
Yes, yes, yes. Because again, so that is a concept from molecular biology.
Yes, ma'am.
Because when transcription is happening, you always have the poly A coding regions at the 3' extremes, right? So that's how.
Okay, ma'am. From my side, I clear with my all doubts.
Okay, what's your background? Like, are you a honest student, or what has your background been
Currently, I am pursuing my BSc and in biotechnology.
in biotechnology, right? And which year are you in?
I'm a second year student. Currently, I'm running with my third semester.
Okay, you're in second year. So have you like read about cell biology, molecular biology? And you must have
Yes,
read about
ma'am.
molecular biology.
Molecular
Okay,
biology in class 12th and cell biology is in my first year and in my 11th standard as well. I've studied.
okay. So I think you must have encountered the words that I keep saying.
Yes, ma'am.
Yeah.
That's what I somewhat got an idea about ER trafficking
Mhm.
that you are trying to explaining me.
Okay. Okay, so if everything is done for today's class, I'll see you in next class. We'll start with our T cells, which is on Tuesday. Okay.
Okay, ma'am. Thank you so much, ma'am.
Okay. Take care.